Hello friends, welcome back to the video. Today we are going to discuss about the plant tissue culture. Okay, this is very important discovery as it has unlocked the complete potential of the plant. Okay, so first of all, what exactly is a plant tissue culture? So plant tissue culture, it can be defined as growing the plant cell, tissues or organ in a sterile, nutrient-rich environment. Okay, so the requirement is that the environment or the media where we are culturing it should be sterile, means lack of contamination. Okay, and it has to be nutrient rich, then only the growth will occur. Okay, so how does like how does the plant tissue culture concept is important and how it happens? So the plant cell, it has a capacity that is known as totipotency. Okay, now, this totipotency potential is exploited when we are talking about the plant tissue culture. Totipotency, it's a potential of the plant cell to, okay, it's a potential of the cell to grow into complete plant. Okay, so most the plant cells, if we are exploiting and we are providing all the nutrients that is required for its proper growth and development, then they will develop in the aseptic conditions also. So the next is why we go for the plant tissue culture. In the nature, there is already the plants are surviving, right? Then why we need to go for the tissue culture? So there are various advantages of I will abbreviate the plant tissue culture as PTC. Okay, so what are the advantages? Why we do? Because it's a rapid propagation. Suppose if you want to grow for cultivation of the tree plants. Okay, suppose we want to go for the cultivation of poplar. So it has very long life cycle. Okay, so what we can do, we can just take any explant. Explant means excised plant part of poplar and then grow it in the culture media. Okay, so it will grow, and then those grown stem, we can do its rooting and then transfer it to the soil. So in that procedure we do not rely on the environmental condition also, and we can produce large number of plants. Okay, so that's why it is the rapid propagation. The second is the disease elimination. Okay, so we can just choose the healthy ones when we are propagating. Okay, then the next is the genetic improvement. The plant tissue culture, it offers the benefit of genetic improvement. It means that we can introduce a gene, okay, to improve the or to get the desirable trait. Okay, and the next advantage is year-round production. It means this is not year, Y-E-A-R, year-round production. So it means that we can grow the plant anytime. Okay, now I will explain you how all this process. These processes are like if there is some endangered plant. Okay, there are many endangered medicinal plants. For example, there is one that is known as pecoriza. Okay, so we can just use the plant part of that endangered medicinal plant and then cultivate it under the tissue culture, and we can multiply it. Okay, second, commercial plants. Okay, there are many commercial attractive plants, okay, that is required in the very high quantity, like there are flowering orchids. Okay, so they are in very high demand and they are very costly also. So the plant tissue culture, it offers ease to multiply it. Okay, so now we are going to learn it the steps of plant tissue culture. Okay, so there are various steps of the plant tissue culture. Okay, so the first step is selection or and the explant preparation. Okay, selection and explant preparation. So what does it mean? First of all, we need to identify that we need to identify, or we need to select that which species we want to multiply using plant tissue culture. Okay, which species and what is its importance. Okay, because it's a aseptic procedure, you know that there is a uh, highly skilled labor is involved. Okay, and we need sophisticated labs also. So while selecting the species, we have to be very clear that why it is important, why it need to undergo the plant tissue culture system, why not the traditional system. Is the plant tissue culture is offering a advantage that lacks in the natural system? So then only you have to proceed, right? So you don't go for cultivating the suppose the mustard plant in the tissue culture. That is not beneficial, right? If you want it for the commercial purpose, then you will sow it in the field condition only, right? So we have to select it, which species we have to do. Okay, why it is important. Then we have to go for explant preparation. So it is asked in the examination that what is explant. Okay, so X stand for excised. Excised means detached or cut plant. Okay, so we can take any tissue, stem, leaf, cotyledons, root tissue also. Okay, so the second step is sterilization. Okay, so now because when we are taking actually the explant from the external condition, right, from the field condition or already growing plants in the pot. Okay, so we need to sterile it, because when we are going to if there is contamination, that will also grow in the media, and before the plant actually survive, the contamination will spoil the media. Okay, so we need to sterile. So usually we are using the bleach. Okay, sodium hypochlorite. Okay, and then we use 70% ethanol also. We are writing ethanol, ETOH usually. Okay, so we are using the ethanol also. So what we will do, we will just either we will dip if the tissue is fragile, right? Or if we are growing, suppose we are taking the seeds, then we will submerge it. Okay, for few minutes. There is mercury chloride also we are using. We will submerge it for some time, so that it can just kill the contaminants, not the actual tissue. Okay, so that's the third step in the process of tissue culture is culturing. Okay, so now uh, we have already selected which species we have to culture, right? We have already excised the desired plant part, and then we have sterilized it. Now what we do next is the culturing. So we will put the explant on media. Okay, so what is media? Like we will grow the explant in two kind of medias, solid and liquid. Okay, depending upon the plant tissue and uh, its property, where it can multiply fast or it can grow fast, right? So the solid media is supplemented with both these media, they are supplemented with the essential nutrient that is required for the plant regeneration. Okay, so we will just I will draw it for you. Usually we are culturing in the tubes or the flask. Okay, so just imagine that this is a flask and this here is the media. We will pour the media. It's a solid media. Okay, so it's like the media is fulfilled here, and we will just keep our explant here. Okay, and we will just cover this with the cotton plug. Okay, so this cotton plug will ensure that there is oxygen is passed, but the contaminants were not. Okay, so it will act as a filter. Okay, so we will put the culture the explant on the media. So after the culturing, the next step is the fourth step. It is the differentiation. Okay, differentiation means now the differentiation. Okay, so now the our grown our cultured plant part, it will start to grow. Okay, So what will happen during the differentiation, that suppose this is I will just draw like this is our media and this is our tissue. So what will happen, that this is the tissue, I will write for you, this is the medium. Okay, media I will write. Okay, now carefully understand that this is the tissue, this is the tissue above and this is the media below. Okay, so now what will happen, that first of all, most of the cases this tissue will give rise to callus. Okay, so what is callus? It's it will grow like in a mass. Okay, with no differentiation, but these are alive. Okay, so sometimes what we do is that we have callus, the same media, the plant will develop the root or shoots. Okay, if not, then what we will do, that we will transfer this callus to a new media. Okay, that has the hormones that can develop the shoot tissue. Okay, so sometimes the roots will also develop here in the same media, and if not, then what we will do, we will just take this individually grown plantlet. Okay, and we will put that in the media that promotes the rooting. Okay, this usually we are doing in the test tubes. Okay, and we are usually using the auxins to promote the rooting. So once this is done, we'll move to our next step, that is the acclimatization. Acclimatization means all this thing is done under a tissue culture system in the lab, right? Now we have to move over these plantlets to the soil. So we will just keep them into the condition before particular condition before transferring it to the field. So what we will do, we will just cover them up. Okay, and they will try to provide the high humidity, and we will expose them to a particular temperature where we are going to transplant it. Okay, and then finally we will transfer it to the soil. Okay, so these are the various steps of plant tissue culture. I hope it is clear to you. One thing I want to mention here, that after culturing the explant, we will move these cultured plants to lab condition. Okay, we have a special labs like that is known as plant tissue culture lab. Okay, and there we are maintaining the essential factors like light, temperature and humidity. Okay, so under control condition they are grown. So next we have to talk about like there are how many types of tissue culture. So if we are talking about the types, then basically there are 4 types of tissue culture. Okay, the first is micropropagation. The next is the callus culture. The third is the anther culture and protoplast culture. Okay, so we have to decide based on what we want. Okay, then we will choose that which kind of tissue culture approach we have to go. Suppose if you want to go for the micropropagation, then our objective must be to make the identical plant from the single source, right? Suppose I have a chrysanthemum. Okay, I want to multiply the same chrysanthemum because it has very attractive color. So I will go for the micropropagation approach. I will just take the explant, any explant from that tissue. I will grow it, and then I will now transfer it to the soil, and then those numerous transplanted plants will produce the desired color flower. Okay, then there is a callus culture. So the aim of the callus culture is to grow the undifferentiated cell, and this is basically used for the genetic manipulations. Okay, and if our purpose is to develop the haploid plant for the breeding purposes, then we'll go for the anther culture. And there is protoplast culture also. So protoplast culture will do for culturing the plant cells without cell wall. Okay, So these are the 4 different types of tissue culture approaches. Okay, now what are the application of tissue culture? So the first application is in horticulture. Okay, so in the horticulture, ornamental plants, they are mass produced. Okay, for example, for the orchids or the gladiolus has very beautiful flowers, so we can go use the tissue culture for the horticultural purposes. Okay, the second is we can use the tissue culture for the agriculture purpose. Okay, for example, if we have turmeric, we know that it is highly resistant to the stress or some environmental variable, then we will culture those rhizome into the tissue culture condition, and then we can develop lots of plant and we can transfer it to the soil. Okay, so in that way we will get the disease resistant high yielding crop. Okay, then the next application is in forestry. Okay, so there are we can go for the conservation, we can also go for the reforestation. Okay, we can develop multiple huge amount of plants and then we can go for their reforestation. We can cultivate them into the forest. Okay, for example, we can grow the teaks or the eucalyptus. Okay, and then the last application we are going to talk in this video is in the pharmaceutical. Okay, so I told you there are many endangered plants that are having anti-cancerous activity or antioxidant activity. Okay, suppose if you are not talking about the endangered, then there is a ginseng. Okay, so that is having quite, it will promote our health. So what we will do, we will just take those explants that are showing the high content of that particular active compound. Suppose only turmeric only, we want curcumin, we want large amount of curcumin, so we can either harvest it from 1 kg turmeric rhizome bio. Okay, and or we can do, what we can do is like we can just take the explants and then we can go for its callus production, and we will make it we will multiply it 1,000 folds, and then we can harvest the curcumin, right? So in that case we will get the higher curcumin content. So tissue culture approach is used also for producing the valuable medicinal compounds. Okay, so I already told you about the advantages of tissue culture. So if we talk about the advantages of the tissue culture in the pointwise manner, then the first is the genetic uniformity. We will get the genetically uniform material. Okay. Then there is a mass scale production. Disease free plant. Okay, conservation and year round cultivation or production. Okay, so these are the advantages. We have discussed it before also, but I wanted to make this point again. Okay, so next we are going to talk about the challenges. Okay, so although it is very fascinating, it has several challenges. First of all is we require high cost equipments. Okay, and we require special facilities. Then we require a skilled labor because it has to be sterile, so the skilled labor is a must for it. Okay, then there is a risk of contamination. Even if you take every precaution, then also sometimes the contaminants which are present in the airs, like suppose spores, it will destroy your culture when we are transferring the explant from one flask to the another one. Or sometimes the contaminant is like endogenous, some fungi is already present in the rhizome tissue and then it will grow, right? So there is a high risk of contamination, and somaclonal variation is also observed. Okay, that these somaclonal variation, they are sometimes desirable and most of the time they are undesirable only. So it may happen. Okay, So now you know all the advantages it offer. uh, okay, and what are its limitation also, what are its uses also. But what's the future, what we see in the tissue culture future? So what we can do is, like, we need very high skill labor, so we can go for the automation of culturing process. Okay, so automation and robotics, it can uh, increase the efficiency, to increase efficiency. Okay, so because then there is tissue culture is offer the potential, right, for space exploration. How we, like, we cannot directly go to the moon or the Mars or any other planet, right? But we, the science has advanced so far that we know what are the environmental conditions there, right? What's the temperature, what kind of nutrient composition is there, and like how far it is from sun, etc., right? Different variables, so we can just stimulate those under the lab condition, and we can see, okay, which plants are performing better, which are not, before actually sending the seeds to the space, right? And the last is, we have to improvise the media for wider application. Okay, so still we need to develop a very um, a media that is improvised and that has a very wide range of, adaptability. It means that like, they usually we are using Mirashigi and Skoog media. Okay, so this is this media, it helps to grow several thousands or lakhs species. Okay, however, we need to play with the hormone, depends upon the tissue condition. But still there is a scope that we can just make a media that is acceptable by the every plant. So more research is needed in this direction. Okay, so I hope I tried to make the point in this media, that why the tissue culture is important, and what are the different type of tissue culture, what are the advantages associated with it, and what are its application, challenges and limit, along with covering the future of tissue culture. Okay, thank you for watching my video.